Good morning, everyone. Today we are going to cover topic 5.4, the human alimentary canal, and this is part 4 of the series. The learning objective is still the same, but today we are going to focus on absorption and egestion only. Assimilation, we're going to do that in another video because I feel like it is a big concept and we probably need one whole lesson for that. Before we start, let's do a recap of last lesson. So we looked into digestion and we actually looked at fully what are the digestive processes that occurs in the human alimentary canal. So we have the mouth or buccal cavity. This is where digestion actually starts. So the mouth, over there we also have mechanical digestion happening, and this is the action of the teeth. And this helps to cut food into smaller pieces, so only physically but not chemically smaller. The reason for cutting food into smaller pieces is really good so that we increase the surface area for enzymes to act on. And that is what we call chemical digestion. Chemical digestion starts in the mouth, and this only occurs with starch. Starch digestion only will occur in the mouth, and the enzyme that does this is amylase, and we get the product maltose. Next up is the stomach. Stomach only involves chemical digestion of protein with the enzyme pepsin, and the result, we get the product short polypeptide chains. The third area of digestion in the human alimentary canal is actually the duodenum. So firstly, we can cover is the mechanical digestion part, and this is actually the action of bile. Bile emulsifies fat. So what this means is that it will convert the large insoluble means that it won't dissolve in water, fat globule into soluble fat droplets. So they become smaller physically, but not chemically. Now let's look into the chemical digestion found in the duodenum. So we have starch digestion occurring there. This is due to the enzyme, the pancreatic amylase, and the product is maltose. Maltose will be further broken down by maltase, which is made by the duodenum wall. And so this is called the intestinal juice. The product that you get from digestion of maltose is glucose plus glucose. So this is actually separated. Now you get individual, very small molecules of glucose. And that is the simplest form of carbohydrates. Sucrose can also be digested in the duodenum. This is a disaccharide and it's made up of one molecule of glucose and one molecule of fructose. And this enzyme sucrase breaks them apart. And this is also the smallest type of carbohydrate. And lastly, for finishing off digestion of carbohydrate, we have digestion of lactose in the duodenum. And this is done by the action of lactase enzyme. And this enzyme breaks down lactose into glucose and galactose, which is also the smallest unit of carbohydrate. Protein digestion also happens in the duodenum. So big protein molecules will be broken down by trypsin, and this is found in the pancreatic juice. So it's made in the pancreas, whereas pepsin is a protease that is made in the stomach. And these two actually, they do pretty similar things, but they have very different pH optimum. For pepsin, because it is in the stomach, so the pH is around 2, it's very low, whereas trypsin works in the duodenum. So they require a more alkaline environment, and thanks to bile, it provides that optimum pH environment. So trypsin breaks down protein into short polypeptide chains, and protein digestion is finished in the duodenum, aka completed, by the action of erepsin, which is found in the intestinal juice, and erupts and breaks down the short polypeptide chains into amino acids. These are individual small protein molecules. Fat digestion also occurs in the duodenum, and this is due to the enzyme lipase, and the product here is fatty acids and glycerol. So fatty acid is actually these three zigzaggy lines here, and glycerol is this one big part right there. Just so you know, fat, another name for it is triglyceride. So the tri comes from 3 fatty acids, and glyceride, I think, comes from glycerol. So that's why when we break down fat in our body, lipase breaks it down into fatty acids and glycerol. Now, after digestion is fully completed in the duodenum, the small intestine, which is the ileum part, is going to carry out absorption. So over here is your end of duodenum, and the chyme here has actually very, very small molecules. So you have amino acids, individual glucose, there's fructose, galactose, and you have glycerol and even fatty acids. So these are very tiny and they are very soluble and can actually be absorbed into individual cells. So that's why you want to break down food, because you need to absorb it into the cells itself. So it needs to be very, very small. So what happens is that the chyme, which contains all the digested products, now will enter the ileum, which is the end part, the last part of the small intestine. So this is where most absorption takes place. Absorption is the process whereby the body takes in digested food and nutrients into the bloodstream. The small intestine absorbs the digested food, which is the digested carbohydrates, digested protein, and digested fat. It also absorbs vitamins, which you don't digest because it's very small already. And it also absorbs water, and you don't digest water either because water is very small as well. So this is a picture of what your small intestine actually looks like, and you can see that there are like blood vessels actually surrounding it, and that actually is related to its absorption function. So here are some real life photos. I'm really sorry that that's pretty graphic, but that is how your small intestine, your ileum, actually looks like. So here is a diagram of what it looks like inside. So you can see here, these are veins, so the blood vessels that you can see, and also arteries. And they're connected to this very important vein called the hepatic portal vein. And this is a group of blood vessels, which we will talk about in another day. And we also have these muscle layers here. So you have your circular muscles, and then on the outside you have your longitudinal muscles, and this will actually carry out peristalsis. And then if you look at the lumen, so the hole here, it's surrounded by these like folded layers. So the small intestine has this like folded layers on the inside, and these folded layers are very important for its function of absorption. To improve its efficiency, the surface area of the ileum is increased in the following ways. So for everybody, you actually have 7 meters long of ileum in your body. So this means that you are going to take a lot of time and be given a lot of time to absorb all the nutrients. The walls are folded or pleated longitudinally, so that also increases surface area for absorption. And the walls have these million microscopic finger-like projection called villi. So this is how it looks like under the microscope, and this is, I think, an electron microscope. And so those things right there is actually the walls right here. And if we draw that in a diagram, it looks kind of like this. So these are the 3 adaptations of the ileum for absorption. The villi is the finger-like structure that we looked at. If you're talking about just one, you call that a villus, but plural, many of them, we call that villi. So this also has its own adaptations for absorption. Number one, it's extremely numerous. It means that there's a lot of them, which increases internal surface area of the ileum. So you can see that's just a small portion, and there's like a lot of those finger-like structures. That's not really the color of it. It's just what people do when they get a electron microscope picture. They can actually add their own colors. So I think this might have been the best color to distinguish each and every one of them, maybe. So just to let you know that the inside of your intestine doesn't really look that brown, I guess. Okay, the second thing is that the villi have very thin walls called epithelium, and this is made up of 1 cell thick epithelial cells. So being 1 cell thick is really good because it means that things can pass through it much more easily. So this is what you call epithelium, which is this whole layer right here. So this is the wall of each, this is a villus, so there's one. So this is the wall of it. And then each one of these little dots, it actually refers to 1 cell. So the wall itself is actually made up of cells, and it's only 1 cell thick. These this villus right here, they contain special structures called lacteals that absorbs fatty acids and glycerol. And another thing is that they also have blood capillaries surrounding the lacteal and actually inside each villus. So this blood capillary is important for transport of substances to all parts of your body. And the fifth is actually each of these guys right here, they can move. So they are able to bring themselves in contact with the food so that they can absorb them even more faster. Here we have a video of the inside of the lumen of the ileum. So it actually shows the folded walls of the ileum, and also these folded walls are even thrown into more folds, which is the villi itself. You can see here that is the lumen space, and it's actually filled with liquid. So this is what happens if we are your chyme and we just got out of the duodenum and now we're inside the small intestine. You can see the finger-like structures right there, and the walls are actually thrown into folds. The ileum itself is moving, and I think right now, as you can see, that the lumen size is getting smaller. That's the contraction going on, and that's actually the contraction of the circular muscle, and then the relaxation of the longitudinal muscle, making the lumen size much more smaller. So we can fast forward this little bit, and you can see that the lumen size is getting much more smaller, and that actually helps to push the food down the digestive tract. Okay, so now let's focus on the absorption part of the villi that can be found in the ileum. So after digestion is complete, the nutrients are now small enough to be absorbed into the epithelial cells that are found here on the surface of the villi. So once you eat like a very big songkai or a very big lunch or dinner, you're going to have a lot of nutrients. You can see here that it's not just 1 or 2 glucose molecules. You actually have like quite a lot of red right here. And they're small enough that they can now pass through each of the cells on the surface of the villus. If we zoom in right here, 1, 2, 3, this is what it's actually going to look like. each epithelial cell also increases the surface area for absorption by having these finger-like structures on them as well, called microvilli. So microvilli can be found on each epithelial cell, and this is just like how the shape of it is. So the cell membrane here is thrown into folds as well. So all the carbohydrates, the proteins, the vitamins, mineral salts, and water will actually pass through the villus and it will enter the blood vessels. Over here on the upper hand here, like on the top, not really the top of everything, but you have two major blood vessels here. The blue color actually refers to a vein, so this is going towards the heart. And then you are going to have this one here too. So this is oxygenated blood that is fresh from the heart and the lungs. So blood acts as a transport system, and all of your carbohydrates, protein, vitamins, mineral salts, and water will actually get absorbed and be transported to all parts of your body by the blood. But how does absorption take place? Absorption takes place by the process of diffusion. So because we have high concentration of nutrients on the outside of the cell membrane of the epithelial cell, then inside what's going to happen is that those nutrients are going to move from a region of higher concentration to a region of lower concentration. So down a concentration gradient, so by diffusion, it's going to enter the epithelial cells. But in the case that there isn't like much outside here, you can actually absorb by active transport. Carbs and protein, again, will enter the blood vessels, but the fat, so the fatty acids and glycerol, will enter in a different way. Fat, aka the fatty acids and glycerol, when it's digested, it will pass through the villus walls as well by diffusion and active transport, but instead it's going into the lacteal. And this lacteal will carry all the fat into what we call lymphatic capillaries. Here we have the epithelial cell. The black color line right there is referring to the cell membrane. And we have the nucleus right here, and over here is the cytoplasm. The finger-like projection is called the microvilli. During absorption, amino acids, galactose, glucose, fructose, and glycerol actually get diffused into the cytoplasm of the epithelial cell because they are soluble in water. However, fatty acids are not soluble in water, therefore they need help from bile. What happens is that the fatty acids will react with bile salts to form these things called soluble soaps. These are soluble, so it will then be able to diffuse into the epithelial cells, therefore into the epithelium. And then over here, what happens is that the glycerol will actually recombine with the soluble soap to form fat globules. So these fat globules will actually be absorbed by the lymphatic capillaries. Now that we have most of the things, or actually all of the things, being absorbed. So once your body absorbs the important nutrients, what's left is actually the stuff that you do not digest. And that is related to the function of the large intestine, rectum, and the anus. So we talked about chyme before, and chyme is this mixture that you get after the bolus actually is released from the stomach. And so since this chyme is going to now move into the colon, and while it moves, so when the colon does the peristalsis and moves this food along, it actually absorbs all of the mineral salts and also excess water. So what happens is that the excess water and minerals, once it's being absorbed, the chyme now becomes more solidified. So now we have feces, and along the way, pigments from broken down red blood cells actually give feces that distinct brown color. Over here we have is the rectum, and down there is the anus, and the undigested, unabsorbed matter called feces will make its way to the rectum, where it's actually temporarily stored here. The feces will then be discharged through the anus, which is the hole right here, and this process is actually called egestion or defecation. So the feces will be stored in the rectum temporarily before egestion takes place. And that is the end of our lesson today.